Hello everyone, welcome to Lab Science Tutorials by Archana. In today's video, we will be talking about the heme metabolism. We will discuss how heme is broken down, how bilirubin travels in the body, and how it is finally excreted. So without much delay, let's get into the video. As we all know, hemoglobin is a conjugated protein. It has heme as the prosthetic group. So the non-protein part of hemoglobin is heme. And the protein part is known as the globin chain. Okay, the protein part is globin chain. Hemoglobin is a tetrameric protein, which means it has 4 subunits. Each subunit contains one prosthetic group, that is the heme group, attached to a globin polypeptide chain. For example, in adult hemoglobin, that is HbA, the subunits are alpha 2 and beta 2, whereas the fetal hemoglobin, that is HbF, the subunits are alpha 2 and gamma 2, and HbA2, which is a minor adult hemoglobin variant, the subunits are alpha 2 and delta 2. So these are the subunits of hemoglobin. Now, heme is not only present in hemoglobin, heme is also present in other compounds such as myoglobin, cytochromes, peroxidase, catalase, tryptophan pyrrolase, and nitric oxide synthase. So these are the compounds where heme is present. And the molecular weight of hemoglobin is about 67,000 Dalton, and each gram of hemoglobin contains 3.4 gram of iron. So iron is also present in hemoglobin. It is actually present inside the heme. Now let's see the structure of heme. Heme is produced by the combination of iron with porphyrin ring. Okay, porphyrin ring. Now what do you mean by porphyrin rings? So porphyrin ring system, it is a cyclic compounds which is formed by the fusion of 4 pyrrole rings linked by methylene bridges. Okay, so basically there are 4 pyrrole rings. So if you see the picture here, so these are the pyrrole rings. So this is the pyrrole ring number 1, pyrrole ring number 2, 3 and 4. These pyrrole rings are bridged by methylene bridges. Okay, alpha, beta, gamma and delta bridges, and other groups are also present here, which is methyl group, vinyl group and propionyl group. So this is a porphyrin structure, and this is the structure of heme. So this is the basic structure of heme. So anyhow, I'm not going to talk more about the synthesis and structure of heme in this video. We will discuss it in another video. I'll be focusing more on the catabolism of heme. So let's start the catabolism of heme, which is our major topic for today's session. We all know that the lifespan of RBC is 120 days. Now, what happens to the RBC after 120 days? These RBCs are taken up by the reticuloendothelial system, and which is mainly in the spleen, and the breakdown takes place. Now, when RBC undergoes breakdown, hemoglobin is released, and then the catabolism of hemoglobin will take place. So, what happens to hemoglobin? Initially, hemoglobin will break into heme and globin. Okay, heme and globin. And the globin part, as you all know, it is a protein part, and proteins are made up of amino acids. So when your globin chain breaks down, you will get amino acid, and that will move into the body's amino acid food. Now, what happens to the heme? Heme is a ring structure. It is a porphyrin ring structure, right? Now, there is an enzyme called hemoglobin oxygenase, and that will open the ring structure of heme, and it will release the iron. Now, what happens to the iron? Iron is released and it is reused by the body. So iron is recycled here. Okay, iron is released and reused by the body. What happens to the remaining portion? This heme will convert into biliverdin, and biliverdin is then converted into bilirubin. So the end product of heme catabolism is bilirubin. Okay, bilirubin is the end product of heme catabolism. As you can see in this picture here, the heme portion is acted by heme oxygenase system. Heme oxygenase system, and that requires oxygen, cytochrome C and NADPH. Oxygen, cytochrome C and NADPH. So this is an important point to be remembered. And this heme oxygenase system will convert heme into biliverdin. And there is an enzyme called biliverdin reductase that will convert biliverdin into bilirubin. So bilirubin is thus formed and it is the end product of heme catabolism. The bilirubin formed here is called as unconjugated bilirubin because it is water insoluble. It is water insoluble, hence it is called unconjugated bilirubin. Okay, now let's see the journey of bilirubin. Okay, let's see the journey of bilirubin. So from where does it takes place, or from where does it starts? It starts from the destruction of RBC. So RBC destruction takes place. Hemoglobin breaks down into heme and globin. The globin part will move into the amino acid pool, and the heme portion will convert into bilirubin. Right now, this is the entire thing is taking place inside the spleen, inside the reticular endothelial system. Now, and the bilirubin formed here is known as unconjugated bilirubin. Now, what happens to the unconjugated bilirubin? The unconjugated bilirubin is then taken up by albumin, right? Okay, albumin, and this albumin will transport unconjugated bilirubin into liver cell. Okay, liver cell. Now why it has to be transported into the liver cell? Because in order to excrete the bilirubin, it has to be conjugated. Okay, it has to be conjugated, and the conjugation will takes place in the liver. Liver is the site of conjugation. Okay, so how does it happens? There is an enzyme called urate glucuronide transferase in the liver. It will convert unconjugated bilirubin into conjugated bilirubin. Okay, so this is happening inside the liver. So bilirubin has now reached liver. From where does it reach? It from the spleen, right? Now what happens to the conjugated bilirubin? Conjugated bilirubin is excreted into the bile, and it reaches the intestine. In the intestine, there are gut bacteria, and this bacteria acts on conjugated bilirubin and it converts it into urobilinogen, right? And about 50 to 250 milligram of urobilinogen is excreted in the fecus. Now, what happens means 80 percentage, around 80 percentage of urobilinogen will then convert into stercobilinogen and stercobilin, and it is excreted in the fecus. So 80 percentage of urobilinogen formed is excreted in the fecus in the form of stercobilin. Now what happens to remaining 20 percentage? The 20 percentage will enter into enterohepatic circulation, and it will absorbed and recirculated into the liver, and the liver will re-excreted it in the fecus. So that what's happening here. So 20 percentage will again go back to the liver, and that will be re-excreted in the fecus, but a small portion of it will enter the systemic circulation. Okay, it will enter the systemic circulation, and that will excreted in the urine. About 1 to 4 milligram of urobilinogen is excreted in the urine. So this is the story of bilirubin. Now I'll explain it once again with the help of this picture. So initially this happens in the reticular endothelial system. In the spleen, RBC will break down, hemoglobin will break down, and heme and globin will be formed, and the heme is converting into bilirubin. So this is happening in the spleen, and from there albumin will takes up the bilirubin and it will reach in the liver. Conjugation will takes place, and through bile it will reaches the intestine. From intestine, what happens? 80 percentage of urobilinogen will convert into stercobilinogen and stercobilin, and this will be excreted in the fecus, whereas remaining 20 percentage will enter enterohepatic circulation. It will go back to liver and it will be re-excreted, but a small portion of it will enter into the circulation, systemic circulation. It will enter in the blood, and then it is excreted in the or it will excreted in the urine. That is 1 to 4 milligram is excreted in the urine. So this is the entire pathway of bilirubin, and this is the journey of bilirubin. Okay, now I hope you all understood the portion, understood how bilirubin is formed and how it is transported, and we will be discussing the diseases associated with it, that is the jaundice, the differential diagnosis of jaundice, and all those things we will be discussing in our next video. So if you like this video, please do like, share and subscribe, and stay tuned for more videos. Thank you so much.